to this new liver seminar. It is for me a great pleasure to present Professor Bart Stiles, who is director of an INSERM unit at the Institut Pasteur of Lille. And he is there the coordinator of the Inter-Organ Crosstalk in Cardiometabolic Diseases team. So Professor Stiles did his PhD in Belgium and then the postdoc at the University of California. And I think that it is such a luxury to have him today because it is one of the most impressive CVs that I have ever seen. The numbers are really amazing. He has published more than 900 papers. He has more than 130,000 citations, and his age index is 184. So these impressive numbers, I think that are according to his research publications, that he did some really breakthroughs in the field. And today he is going to present the talk about pathophysiological mechanisms and regulation of metabolic dysfunction associated steatotic liver disease. So all the questions that are regarding this talk, you can write them in the chat and I will transmit them to Professor Stiles. And I think that whenever you want, Bart, the floor is yours.
Thank you, Ruben, for this very nice introduction. It always reminds me of how old I am, because all these numbers, of course, count up when you get older and older. But anyway, thank you for mentioning them. So what I'll do today is give you sort of like an overview of research that we're doing in my laboratory in Lille, which goes actually from bench to bedside and back. As you will see, we do quite a bit of translational research in patients over the last years. And I think this is, of course, in addition to mechanistic studies, very important with respect to potential therapeutic applications. And I'll finish my talk with some of the latest news on that aspect as well. So I don't think that I have to explain to this audience what non-alcoholic fatty liver disease is. You all know that it starts with steatosis, liver fat accumulation, and then progresses to steatohepatitis. The hallmark of NAFLD is, of course, the presence of steatosis. But from a treatment viewpoint, if we look at the endpoints of current running clinical trials, steatosis is not included. So more important are the next steps, such as the development of steatohepatitis and fibrosis, because the trajectory of the patients is such that these evolutions of the liver phenotype puts them at higher risk for outcomes which can be liver-related outcomes such as cirrhosis and hepatocellular carcinoma, but can be also touching other organs such as the heart, because we know that patients with NAFLD have a high risk to develop cardiovascular diseases, which is one of the topics of interest also in my laboratory. Now, I'm sure you all heard about the change in nomenclature where non-alcoholic fatty liver was changed into steatotic liver disease, and NAFLD became metabolic dysfunction associated steatotic liver disease. And I won't go into a discussion of nomenclature. I've not been involved in this. I think it makes sense. But I would like to mention that with respect to my talk, the NAFLD and MASLD definitions are identical in the cohorts we study because most of the patients that we study are obese to start with. And similarly, NASH has become MASH, and one of the specific aspects of this nomenclature change is that MASLD patients have to have at least one of the 5 criteria of the metabolic syndrome, in which waist circumference is an indicator of obesity. And therefore, in obese cohorts, you are very quickly in an, I would say, logical, identical definition of NAFLD and MASLD. Now, as I mentioned, it is the NASH stage, and particularly what we now call at-risk NASH, which is currently targeted with respect to therapeutic approaches to treat patients with MASLD. And I'm sure you're all familiar with steatohepatitis being characterized by ballooning of hepatocytes and the appearance of inflammation on top of course, the presence of steatosis, which is, of course, the initial characteristic, as I just mentioned. And when this is combined with early stage fibrosis, F1 to F3 fibrosis, one talks about at-risk NASH, and this is the condition that is now being studied through biopsy measured changes in at-risk NASH in patient populations. Now, with respect to steatosis and fibrosis, we do have a number of non-invasive technologies such as MRI or fibroscans that can help the clinician to have an idea of the liver status of the patient he has in front of him. But for at-risk NASH, there is actually at this time very little available, and so all the studies have to perform pre- and post-treatment biopsies of the patients. And so we were very interested, together with Genfit, which is a company that I co-founded about 25 years ago, and which became very active in NASH around 2013-14. We were interested in identifying a biomarker test, so a non-invasive test, to identify at-risk NASH. And so using the ResolveIt cohort, and some of you might remember that the ResolveIt cohort is the study in which elafibranor, a PPAR alpha delta agonist, was tested for its effect on at-risk NASH. Unfortunately, the study turned out to be not achieving its endpoint and was therefore stopped. But the cohort patients were used to identify parameters such as microRNA, YKL-40, and so on, to develop a test which is called now NIS2+, an evolution of the initial test, which had 4 parameters in it, and therefore was called NIS4. And this test has been now assessed in a large number of studies, so not only in ResolveIt, and shown to have a predictive value for the presence of at-risk NASH, diagnostic value, which is clearly higher than the currently available tests. And this was also shown in a study published by the NIMBLE Consortium in Nature Medicine last year, in which Arun Sanyal and collaborators compared, as we did before here, this test against established tests, and found that it is the most powerful test to diagnose at-risk NASH. And so we've now published a number of papers together with our clinical colleagues showing that the use of this non-invasive test can improve the diagnosis performance. It can be used to increase the the enrollment in NASH clinical studies, because there are a lot of false or negative biopsies when patients are enrolled. And it's also unlike, for instance, tests like FIB4, independent of age, also independent of type 2 diabetes and sex. So it's actually quite an interesting test, and I wanted to mention this to you because I don't think it has been, except for the publications, taken up by the field at this time. Now, for the rest of my talk, I'll go into some of the studies we have been doing using two different cohorts. The first is the RESOLVE NAFLD cohort, which shown on this slide is a study which we started in a long time ago in the HEPADIP FP7 consortium in the 2000s with Sven Franke and Luc van Gaal in Antwerp, who collected biopsies from patients that were referred to the clinic for obesity. And as you can see, the NAS activity scores, NAFLD activity scores are quite surprisingly high in this cohort, which was, of course, by Sven Franke, pre-screened based on known markers such as ASAT and ALAT. So this study is very interesting because the patients have been either undergoing a bariatric surgery or lifestyle intervention, have been followed up, and to the best of my knowledge, it's the only study where quite extensive second biopsies have been collected, not only for diagnosis, but also for research purposes. And so using this study, we did a first transcriptomic analysis comparing the transcriptomes of patients with fatty liver only, NASH without fibrosis, and NASH plus fibrosis. And doing this was done by my colleague Philippe Lefebvre in the lab. We do find a number of pathways that are activated when the disease progresses from fatty liver to steatohepatitis and to fibrosis. We did the analysis cross-sectionally. It's not a longitudinal analysis because in the same patient, biopsies progressing from the same patient, disease progression biopsies from the same patient are unavailable anywhere in the world. The interesting part is, I think, except for the classical pathways of lipid metabolism in the fatty liver stage, inflammation, ballooning, in inflammatory responses and PGFR signaling pathways in the NASH stage, and then epithelial mesenchymal transition in the fibrosis stages, pathways you would expect have been reproduced by other studies. But the interesting part is that the transcriptomes of the next stage are already visible in the previous stage, and that suggests that the disease is indeed a progressive disease, an issue which was a discussion at a given time. So what do we think nowadays about this progression of fatty liver to steatohepatitis and the mechanism? Of course, if we talk about MASLD, then the metabolic component becomes even more important. And a number of studies have shown that the progression to fatty liver, from fatty liver to NASH, is actually an incapacity of the liver to further adapt its metabolism due to the environment, the fat accumulation, to such a stage that there is a development of oxidative and ER stress and an impaired respiratory capacity. And that is a hallmark of the metabolic alterations that lead to steatohepatitis, and also the inflammatory response that is observed in this stage. And I'll come back to these mechanisms at the end of my talk. Now, another approach that we did, instead of comparing classical transcriptome analysis, between the different patients in this cohort. Another approach we did with Joel Haas, Luisa Vongia, and Denis Movilenko in the lab was to apply on the whole cohort a WGCNA approach. So actually to identify clusters of co-regulated genes, which then were put in modules and given a color. So using that, we then asked the question, are there any clusters that associate with the liver stage and that also are modulated when the patient has a response to intervention, which can be Roux-en-Y gastric bypass or lifestyle intervention, as I mentioned earlier, and does not respond in LSI non-responders. And so as you can see, we see here a blue cluster in which the genes are generally downregulated when the patients respond to either Roux-en-Y gastric bypass, which most patients respond to with respect to NASH status, as you probably know, but also the lifestyle intervention responders, whereas the non-responders do not respond. So quite interestingly, these responses were independent of changes of BMI in these patient groups. So we're quite interested to find out which genes are represented in this blue module. And somehow, maybe not so surprising, when we looked at these genes, we found that a lot of these genes are related, you can see, to inflammatory pathways. And as I mentioned in the beginning of my talk, of course, the transition of fatty liver to steatohepatitis is ultimately strongly characterized by the appearance of a strong inflammatory response, and you see that quite clearly. And what's interesting is, of course, that intervention that makes the NASH go away also decreases the expression of these genes. Now, this brings me to the whole immune inflammatory component of at-risk NASH. And so with Luisa Vongia and Sven Franke, we went further and collected also blood from patients with histologically diagnosed steatohepatitis with or without type 2 diabetes. And then we did a deep immune phenotyping. As you can see here, we measured quite a large number of immune parameters, which you can only do on fresh material. So we had to organize this in quite a complex manner because Louisa works in Antwerp and we work in Lille, so that's about 2 hours by car if there's no traffic jam. So you can imagine that it was not so easy. What's interesting is that we found a number of inflammatory cell types that clearly associate with stages of histological stages of NASH, as you see here on the bottom. And some of them also correlated with parameters of glucose metabolism, but not all. And so if we look at the circulating immune cell populations, we find a number of cells that we expected, sort of based on literature, such as inflammatory monocytes and Th1 and Th17 cells. We also found less expected cells to be increased, such as CDC2 cells with a decrease of the CDC1 cells in the regulatory T cells. And also very interestingly, find an increase of cytotoxic CD8 T cells, which have also been found to play a role in the transition of NASH to hepatocellular carcinoma. So this is in circulation. We also looked in the liver and we could find several of these cell types also elevated in liver, and we're now currently studying them at a more detailed level using single-cell RNA-seq and other approaches. But that will be for another talk. So one of the questions then is, how does the metabolic environment interact with the immune cells? And as I mentioned, we were quite interested in the dendritic cells. And so in a paper published by Dennis with David Dombrovich, immunologist in my department, we could show that immune cells such as dendritic cells show a combined response to inflammatory signaling, which of course occurs at the stage of NASH through, for instance, toll-like receptor activation, and the exposure to fatty acids, which is often the case under conditions of obesity, as you all know. And what they showed is that the combination of free fatty acids with toll-like receptor ligands, such as shown here, resulted in a strong increase of the inflammatory response of a number of cytokines, such as interleukin-23A. And this induction was due to a change in metabolism that occurs in the cells with alteration in glycolysis that then fed into, again, these pathways that I mentioned at the beginning of my talk, namely oxidative stress and an increase in the unfolded protein response. So this is, I think, very interesting to see that metabolic factors and nutrition-derived components such as fatty acids can interact with for instance, toll-like receptor agonists to induce a stronger immune inflammatory response. So we are working on that in more detail, and hopefully next time I can give you more information from our work. We've also been studying another cohort, which is the ABOS cohort developed by my colleague François Patou in Lille, and this is a very large cohort. It's now more than 1,500 patients, but at the time we did this study with Jimmy Vandel in the lab, we were able to do transcriptomic analysis in a little more than 900 patients. And this ABOS cohort is actually a morbid obese patient cohort, which are patients that undergo bariatric surgery. And so we can obtain a liver biopsy at the time of the surgery. You see the distribution between men and women of healthy, obese, fatty liver, and NASH in this cohort. And it's not a surprise to see that you have a lot of women with a lot of fatty liver, not necessarily NASH, but there's also quite a bit of NASH patients in this group. But very intriguing, and we're looking at those patients in more detail. If you look at individuals with morbid obesity, meaning BMI 35 plus, you do see quite a bit of healthy livers, meaning no steatosis, no NASH, nothing in that sense, especially in the women. So very interesting population, which I think has been understudied so far. So we use this cohort to look at using bootstrapping and random forest bioinformatic approaches to identify which parameters result in the highest heterogeneity in this whole cohort, in the transcriptomic of this whole cohort, if you put them all together. And we were very surprised because, as you can see on this slide, whereas histology is an important factor of heterogeneity, because, as I showed you, we have all the groups of the NAFLD spectrum in this cohort. But very interesting, the most important factor was sex. So it makes a big difference if you look at transcription, genes expression, and transcriptome analysis, whether you look in male or female subjects. And we went a little bit further in that study and looked at what might be the differences between these women and men that explain why this is such a high high parameter for variation of the transcriptome in human liver. And so we found that the pathways are quite different with female livers, meaning more cell-cell contact and immune cell migration, inflammation, extracellular remodeling being altered, whereas in male livers, it's more metabolism and inflammation. So this, I think, is quite an important observation, and should spawn studies to include, and also preclinical studies in mouse models for those who do these studies in their laboratories, to study also the female counterpart in their groups. And another example that I want to show you is a study we did with Guillaume Gridge on this population, and this was done also on the RESOLVE cohort, in which we looked at different metabolic parameters such as branched-chain amino acids, urea cycle components, TCA and glutathione in circulation. And we're doing this now also in the liver, as many researchers are doing, of course. And you find a lot of studies, I cite here a couple of them, that say, well, NASH presence is associated with BCAA elevation, or TCA cycle metabolites alterations are associated with NASH, and so on. But if you look in detail at all these studies, none of them stratify their patient population on age, BMI, HOMA-IR as a measure of insulin resistance, HbA1c, or sex. It's quite surprising, because we know, for instance, that NASH co-segregates quite often with type 2 diabetes. And so if you don't stratify for that, you cannot conclude that whatever you measure is really related to the NASH component. And so I'll show you how you can get into wrong conclusions. We measured the branched-chain amino acids in our cohort, and then we looked using different approaches with which parameters the branched-chain amino acids correlate, or which parameters impact most on these branched-chain amino acids in blood. And to our surprise, it is sex that comes out as number one, much stronger than the NAFLD stage, or, for instance, insulin resistance. And so if you then start correlating these three different branched-chain amino acids in men and women with different stages of NAFLD, you find actually very surprising that the correlation, positive correlation, is strongest in females for almost all the histological components. But this is not the case in the males, and it actually correlates rather negatively in the males. So if you study these branched-chain amino acids and you don't take sex into account, then your result will be very much influenced by the distribution of male and female participants in your study. And another point that we studied, the paper just came out, is Manu Johans in the lab. We did this with Philippe Lefebvre, is whether the transcriptome and metabolome of NAFLD patients is impacted by the time of the day, because we know that the circadian clock plays an important role in liver metabolism and gene expression regulations. And so we did a study where we identified the daytime of sampling of the liver sample. And then we looked whether we could see variation in certain gene expressions, such as the clock genes, between the morning and the afternoon sampling, which we found. And then we looked at pathways that alter also between morning and afternoon, and whether these pathways were impacted by the histological stage of the livers, namely normal versus steatosis versus NASH. And it's quite interesting that you find pathways that are altered in a time of day specific manner and impacted by the liver stage. So the message here is that one has also to take into account what time of the day one takes a sample and compares it, for instance, to the metabolome or the transcriptome of other samples. You can read the details in this paper, it just came out. So to finish a little bit about the therapeutics, and this is already a quite old slide, but it shows you that the landscape of NASH drug discovery has been extremely busy, with drugs going from phase 1, phase 2, and phase 3. And as you know, we now have at least one, finally one drug that made it in phase 3, and I'll comment on that in a minute. Now, there's been many failures. It's not an easy journey. It's been, for different reasons, extremely complicated. Different targets have failed in the clinical studies, amongst which elafibranor, that I worked on quite a bit. It's a RESOLVE trial that I mentioned for development of NIS2. So if you look at what works until, let's say, quite recently, well, bariatric surgery works very well. And this is the study that I mentioned, the RESOLVE study, where we have follow-up biopsies. And these are the so-called the diet group. Remember the lifestyle intervention that I showed for the immune phenotyping. And you see that lifestyle intervention in a very specialized clinic run by Luc van Gaal can decrease the NAS score in the patients. You can see they do improve by this, but it's a very difficult task. And if you look at bariatric surgery, it basically wipes out NAFLD in these patients. They go back, almost all go back to score 0. So it's very, very efficient. And so one of the questions that I've been intrigued with for many, many years is how this works and how Roux-en-Y gastric bypass impacts on lipid metabolism, and specifically on cholesterol metabolism, sterol metabolism. And we've finally finished the paper published by Fanny in the lab last year, where we could come up, again, translational in humans, so mechanistic proof is very difficult to obtain, but where we found evidence that there is a improvement in hepatic function after Roux-en-Y gastric bypass and an alteration of intrahepatic sterol metabolism through the bile acid synthesis pathways. But unlike some of the FXR drugs that work mainly on the classical pathway, we found that in these patients, the alternate pathway via 27-alpha hydroxylase is mainly activated, and this allows the body to eliminate cholesterol through bile acid synthesis into the bile. And this is what you see also in induction of ABC, G5, G8, all pushing the cholesterol coming from the periphery after the Roux-en-Y gastric bypass. There is a strong weight loss, and so the body has to handle not only the triglyceride, but also the cholesterol. Whereas the classical pathway of LDL receptor uptake was rather downregulated 1 year after Roux-en-Y gastric bypass. But in these patients, we also looked at parameters that correlate, metabolic parameters, with the regression of NAFLD. And so what we found is that the changes in parameters that determine bile acids, such as C4, which is a marker of the classical pathway of bile acid synthesis, and FGF19 that blocks the classical pathway, that these correlate positively, respectively, negatively with NASH histology. So again, pointing to a link of this whole cholesterol bile acid pathways and fluxes with changes also in NASH histology after Roux-en-Y gastric bypass. And very interesting is that we also found that there is a strong decrease in markers that are related to de novo hepatic cholesterol synthesis, such as lanosterol and desmosterol, and the decrease correlates with a decrease in basically all the parameters that are used to do the histological analysis. And there's also a decrease in latosterol that is an indicator of intestinal cholesterol absorption. So I think even though I cannot prove this causally in such a study, as you all understand, it indicates that there is an important component in the NAFLD spectrum that is related to the cholesterol bile acid metabolism pathways, but in a quite complex way. So why did we have so many failures until recently? Well, NAFLD is complex, heterogeneous, multifactorial, multi-mechanism disease. And it is in general, and that's work by Jérôme Écoute in my lab, associated with a loss of what we call liver identity. And I'd like to finish on that, coming back to the stressors that are associated with the transition of fatty liver to NASH, namely inflammation, but also endoplasmic reticulum stress, or UPR, as I showed you also previously. And so what Vanessa Dubois, postdoc in the lab with Jérôme and me, showed is that under conditions of liver hepatocyte stress, there is an induction mainly of stress-related genes, as you would expect, with a downregulation of more liver identity genes. And so that led us to propose the concept that under condition of stress, the hepatocyte reacts by a shutdown of its liver identity profile. And this shutdown is due to the fact that there is actually a downregulation of a whole set transcription factors, which are very enriched in the liver and therefore are called liver ID transcription factors. And these factors are thought to work in concert to allow the hepatocyte to exert its function and have its hepatic hepatocyte phenotype. And in this set, you find, of course, classical nuclear receptors like HNF4, but you find also some PPARs, FXR is in there, and so on, SHREP, for instance. So there is a decommissioning of these transcription factors under conditions of hepatocyte stress, and that leads then to a inhibition of the expression of liver ID genes. And what is interesting is you see this switch from liver ID genes down to increase of ER stress genes in conditions of acute liver damage. So we tested this, for instance, in drug-induced injury models or in sepsis models, which is very acute. But you see it also in conditions of chronic liver damage, such as here, related to NAFLD and fibrosis. So it happens also under chronic insults. And the problem of this is that when the healthy liver responds to this aggressor, by this change, this is important to sort of react to the aggressor. But the problem is when this insult becomes a chronic insult, and there is, of course, a long-term loss of hepatocyte identity, and then, as you can imagine, the function of the organ will be compromised. And that's sort of the concept that we are currently further working on. We've shown that this is actually true in humans as well. This is a study done with a colleague of mine in Leuven, and who had livers in the intensive care unit of patients who died there because of critical illness, and compared them to biopsies from healthy individuals. we found a very strong decrease in the PPAR alpha expression and target genes of PPAR alpha in these critically ill livers. And quite interestingly, we correlated this PPAR alpha RNA changes in these individual livers with the metabolic regulated genes much more than with the inflammatory genes. So indicating that under these conditions in this critically ill liver, there is indeed a downregulation of HepID transcription factors, in this case PPAR alpha, and we haven't looked at other ones, unfortunately. And this leads also to a decommissioning of the expression of important metabolic factors. And so this is exactly what we see also when we look in this study in the progression from fatty liver to NASH plus fibrosis. We see a downregulation of PPAR alpha. And we're studying the mechanisms in addition to what I already told you in more detail. But this could be one of the reasons why single PPAR alpha or hepato-only PPAR agonists have not yielded the expected results, because we come in the patients when the pathway is already quite a bit downregulated. The question then is, can you restore the hepatocyte identity and liver function in a condition of hepatocyte damage? And so Julie de Chevalier and Jérôme Écoute worked a little bit more on that, and they actually showed that this HepID transcription factor network that you see here in its complex interaction does actually connect to other transcription factors with less hepatocyte specificity, and they call them the HepID connect transcription factors. And so the idea is that these can cooperate with the HepID transcription factors to increase, again, hepatocyte identity gene expression. Now, quite interestingly, I'm sure most of you have followed the recent literature and communications, is that the nuclear receptor, the thyroid hormone receptor beta, is in this HepID connect transcription factor network. And what we shown in this paper, which I think meanwhile has published, is that when we activate the THR beta factor nuclear receptor in these hepatocytes, that this leads to a reappearance of classical HepID transcription factors such as HNF4, FOXA2, and FXR. So this seems that activating thyroid hormone receptor beta with an agonist can also restore HepID functions. And of course, this brings me to the recent news from earlier this week or last week, it was March 14, that resmetirom, which is a hepatocyte-targeted TR beta agonist, which has shown to achieve the endpoints that were studied in at-risk NASH patients, as I showed you, as I discussed in the beginning, namely NASH resolution without worsening of fibrosis, or fibrosis improvement without worsening of NAS. They achieved this with high statistical significance, and the compound is being FDA accepted last week as DIFRA will be the marketed form. But there are other TR beta agonists. I just showed you one here from Viking that has also results that are in the same line. So it indicates that TR beta agonist is not only resmetirom, but TR beta activation is probably a good way to restore liver functions. And these TR beta agonists also, in addition to impacting on, for instance, liver fat and NASH, also has interesting impact on, for instance, lipoprotein metabolism by decreasing LDL cholesterol and ApoB and triglycerides and even Lp(a). And so this gives us the hope that this drug will also tackle the increased cardiovascular risk that is present in patients with NASH, as I mentioned in the beginning of my talk. There are other compounds, and my lab is mainly working on nuclear receptors, as you probably know. So just show you the results with other nuclear receptor agonists. It's much more complex is the REGENERATE study, which showed positive effects of obeticholic acid in the improvement of fibrosis, but the NASH resolution was much less clear. Finally, FDA did not approve this. The side effects, such as an increase in LDL cholesterol, in contrast to the thyroid hormone receptor beta agonists, as I just mentioned. So that is, of course, a problem for cardiovascular risk. And then finally, there is a pan-PPAR agonist, and so that hits both alpha, beta, and gamma, that has shown quite interesting results on in a phase 2 trial on the criteria recommended by the FDA to study NASH efficacy. And so one of the reasons that I anticipate why this works is because the gamma component on adipose tissue is likely important to lower the free fatty acid return to the liver, which then, as I showed you earlier, can activate the immune cells and do other things, such as leading to steatosis. But it's, of course, speculation, but it could be an explanation why a triple agonist works. So with this, I am at the end of my talk. I think I mentioned every important person contributing to the different studies from the lab in Lille that I have presented. We worked a lot with Antwerp on the RESOLVE cohort with Sven Franken, Luc van Gaal, and colleagues, as well as with François Patou and the hepatologists such as Laurent Dubuquois and Jean-François Goossens for bile acid measurements in the University of Lille, and with Caroline Gauthier, who is specialist of thyroid hormone receptor function. So with this, I thank you for your attention. Here are some of our fundings, and I'd be happy to answer any questions you may have. Thank you for your attention. For reimbursement, the test needs to be FDA approved and probably EMA approved. And so currently, I would say a distributor of the test has been identified. It's LabCorp. GenFit is too small to invest into diagnostic tests. So this is now in the hands of a big company specialized in biomarkers. And of course, it's up to them to bring all these data to the regulatory authorities for approval. We hope that this will be done quite soon. I can tell you, discussing with many of the clinical hepatologists that are involved in all these studies, that I showed the publications, Arun Sunyal, Vlad Radziu, Steve Harrison, Quentin Anstey, and so on, they all would like to test very much NIS2 in their population, so they're waiting eagerly for the access to the test. Yes, of course, it's a balance between the different metabolites that impact on the final response of a given cell type. So yes, I would say that's the case, but we have not studied, in my lab, we have not studied that in much detail. But as I mentioned, there's a big correlation between NASH and type 2 diabetes. So in addition to the lipid alterations in these patients, you also get changes in glucose exposure. So it's likely to be very complicated. Yeah, I didn't mention this because my lab focuses more on the nuclear receptor aspect, but you're absolutely right. There are really exciting data with the GLP-1 injectable analogs. with semaglutide, and as I think Eli Lilly just announced, all these new data come via press releases, and the papers arrive much later, so you have to read what the press releases are to know some of the details. But it looks like tirzepatide, which is a GLP-1 GIP receptor agonist, dual agonist, has even better results with a 70% decrease in NASH in clinical studies. So really exciting data. And then there is also the triple agonists, or these injectables that also act on the glucagon receptor, which from my perspective, I think is more interesting, because I believe that the effects of these single and dual agonists are mainly acting through the brain and decreasing body weight, and that the effect on the liver is a consequence of that phenomenon, a little bit like the Roux-en-Y gastric bypass, which works also. But the triple agonist with the glucagon in there has potential to hit the liver directly, because the glucagon receptor is expressed in the hepatocyte. So this is really interesting, but it's still early days, very exciting. And yes, you're right. Now we have one drug that is FDA approved, resmetirom. I assume that this will come on the market. It depends, of course, on negotiations with the reimbursing insurance companies in the US. The system is quite different, as for instance in France, as you all know. But it should come on the market, and then we'll have to wait for the GLP-1 analogs. Some people predict that these new GLP-1 GIP glucagon receptor analogs are so strong that they will take over the entire treatment of NAFLD. The question is, of course, would they be as efficient on later stage, like fibrotic and cirrhotic patients? That is something we don't know. But we don't know that for the TR-beta agonists either. So there's a lot to be done. But yes, I think you're right. We could come to combination therapy, as we see in diabetes, in hypertension, in dyslipidemia. So finally, we have a drug that made it through the phase 3 trials. That's very, very promising. And we can go now further, have something for the patients, because the graveyard of drugs that failed for different reasons was quite strong, and people were getting very pessimistic. So I think this TR-beta result is now very good news. I think, I mean, I've worked a lot in the lab on endogenous GLP-1. We have done a lot of work on TGR5 that regulates it. And honestly, my conclusion is that it doesn't do much to diabetes. We've tested on the NASH models. It doesn't do anything. But if you inject liraglutide, for instance, it works quite nicely. And so we showed with Vincent Prévost that liraglutide requires passing the blood-brain barrier through the tanycytes. If you knock out the GLP-1 receptor in tanycytes, it doesn't work anymore. So I think these analogs, maybe the ones that touch glucagon receptor are different, do their effect by the strong decrease in body weight and appetite. And in a way, it's very comparable to Roux-en-Y. So I would expect that it would be very similar, but like always in science, we don't know until we do the experiment, right? Very interesting. You're absolutely right. When we look at these type of data sets, of course, we focus on aspects that are allowing us to proceed with our hypothesis, but there are many other indications of other pathways that are regulated when you do this type of analysis. And I agree with you that this is very important. The whole pathways of translation from RNA to ribosomes and so on is more and more appearing as a understudied pathway in metabolic control. I know some people are now looking at RNA ribosome interactions at the whole genome level. So this is a very new field. We haven't studied it in my laboratory, but I think we should, of course, go beyond transcription and go much further. And so, yeah, the fact that they pop up is very intriguing, and so should try to get further with that, or a lab that's specialized in it. I think so, yes. I'll answer you with a study we did with Hervé Guillou in Toulouse. Actually, he did it, and we contributed some ideas to it. It is a preclinical study in mouse models, but for instance, in that study showed what I have known for 40 years, that male and female mice respond differently to PPAR alpha agonists. When I started as a student in the 80s, people were studying peroxisome proliferation, and that's why PPARs were called PPAR, peroxisome proliferation activated receptor. So proliferator activated receptor. So, and we already knew that if we give these drugs to female mice, we get a much lesser response than to males. So yes, there are pathways that are very much sex different. And for those who have listened to Adriana Maggi's talks and presentations, Adriana worked on ER function in hepatocytes, and she has done really, really beautiful work showing how the female liver has a totally different metabolic activity, which is probably related to the fact that it has to gear up its metabolism for during periods of pregnancy. So I think we need to take that into account, and we need to study in all our clinical populations the response of the two sexes and see whether the drugs do as good in each sex and which one is more active in which condition and which sex. It's again a very good point. The fat distribution and then the distribution between subcutaneous and visceral fat is very well known in the field of metabolism to be different, impacting on metabolism. And for instance, in studies were done in the days of the glitazones, the PPAR gamma agonist that acts specifically on adipose tissue. There were clear studies showing different responses to PPAR gamma agonists depending on the origin of the fat, but not only in mice, but also some studies in humans. So yes, because I really think that the metabolic aspect of MASLD, which I think is the good thing of the change in nomenclature, that we put more attention to the metabolic component and less to this non-alcoholic negative definition, that this metabolic condition is really important. And so fatty acid fluxes to the liver from different adipose depots, in my opinion, are really important also. So I think that's definitely the case. How to study that? It's quite complicated. You can do studies in preclinical models, but again, cycles are not the same in mice as in humans, so difficult. If you want to do it in human studies, you need to have very specialized teams like Sam Collins's team who can really measure some of these parameters. It's high-end physiology studies that are required.
Okay, thank you so much, Bart, for the great discussion. Thank you all the attendees for the questions. I think that we can leave it here, and hope to see you all in the next LIBER seminar.